Hi, 안녕하세요. Hi, good afternoon, everyone. This is Hui Foon from GenScript. Thanks for taking time to attend today's webinar. I'm the product manager who is in charge of the GenScript Apex downstream service, which include recombinant proteins, reagent antibodies, assay cell line, and virus vector packaging. It is my pleasure to be here to share the key parameters and tips for ELISA kit development. In today's webinar, I will first talk about what is ELISA kits and the principles and the applications of ELISA kits. Next, I will discuss some of the key parameters and optimization tips for ELISA kit development. Lastly, I will discuss and show what are the capabilities of our GenScript customized ELISA kit development. The enzyme-linked immunosorbent assay, which normally called ELISA, is a commonly used analytical biochemistry assay, first described by Eva and Peter in 1971. The assay uses a solid phase type of enzyme immunoassay to detect the presence of ligands in the liquid sample using antibodies directed against the protein to be measured. It is a common binding assay that allows for sensitive, specific detection and quantitative of target molecules. It is not only used to detect the presence or absence of target molecules in complex biology solution, but also often used to validate specificity of an antibody towards a specific antigen. It is widely used across the broad range of the industrial, such as diagnostic tools in medicines, quality control in food industrial, or validation tools in the basic research. The most commonly used enzyme labels are horseshoes peroxidase and alkaline phosphatase. Other enzymes has been used as well, such as beta-galactosilase, acetylcholine estolase, and catalase. A large selections of substrates are available commercially for performing ELISA with an HRP or AP conjugated. The choice of the substrate depends on the required assay sensitivity and the instrument available for signal detections. ELISA works on the principle that specific antibodies bind to the target antigens and detect the presence and quantity of the antigen binding. In order to increase the sensitivity and precision of the assay, the plate must be coated with antibodies with high affinity. ELISA can provide a useful measurements of antigens antibody concentrations. There are several different types of ELISA. Here I will discuss the common ELISA we normally use. First, this is the direct ELISA. Antigens is immobile on the plate and primary antibody is conjugated to an enzyme bind to the antigens. After that, incubate with an enzyme substrate. There is a colorimetric change. The more antigen in the sample, you can see the color will also become more. In the indirect ELISA, it is the same principle as direct ELISA, but for this kind of ELISA, secondary antibodies is conjugated with an enzyme instead of primary antibodies. In the sandwich ELISA formats, it requires a match, ideally non-overlapping antibodies pair that recognize different epitopes on the same antigens. And this kind of formats is very useful for recognizing, detecting, and quantifying antigens in unknown sample. Sandwich ELISA is generally more sensitive than the direct or indirect ELISA, and it allows for rapid, specific detections of an antigens. Lastly, competitive ELISA is also known as inhibitor ELISA. These formats allow for measurements of an antigen by detecting signal interference where the sample antigens compete with reference antigens for binding to a specific amount of labeled antibodies. The reference antigen is pre-coded on a multiple well. The samples is pre-incubated with labeled antibodies and added to the wells. Depending on the amounts of antigens in the sample, more or less free antibodies will be available to bind the reference antigens. This means the more antibody there is in the sample, the less reference antigens will be detected and the weaker signal will be shown. So some competitive ELISA kits use label antigen instead of a label antibody. The label antigens and the unlabeled sample antigens compete for binding to the primary antibodies. The lower the amount of the antigens in the sample, the stronger the signal due to more label antigens in the well. So this is a summary of the pros and cons of each type of ELISA's format. For direct ELISA type, the procedure is simple, less time consuming and less reagent required. It is also less chances of cross-reactivity due to it is only one primary antibody needed. However, the disadvantage will be the antigen immobile is not specific due to the background noise issue. It is only rely on the primary antibodies, which is less flexible and less sensitivity due to the absence of the signal amplifications. For the indirect ELISA type, there are secondary antibody involved, therefore higher signal amplifications and flexibility. However, the cross-reactivity might occur and longer operation procedure. For the sandwich ELISA, this kind of ELISA offer a high sensitivity, specificity compared to the direct and indirect ELISA. It can recognize different epitopes from one antigens and it can differentiate antigen from unknown sample. However, in terms of the pricing wise, it is more expensive and it is time consuming. Sometimes it is hard to generate a strong antibody pairs against one antigen. For the competitive ELISA, it is useful to quantify antigens in complex samples. It does not require antibody pairs. However, the weakness of this type of ELISA is always require a standard curve to verify the outcome, and sometimes it is hard to measure and require multiple step process. So ELISA kits is widely used in many aspects, for example, in vitro diagnostic, therapeutic drug developments, agriculture diagnostics, and animals and plants disease diagnostic, and also in the food safety. In the IVD area, it is used to detect and quantify the antigens, antibodies, hormones, and other molecules. For example, pregnancy kits, it is one kind of the example. In the therapeutic drug development area, examine the drug absorption, distribution, metabolism, excretions, immunoresponse after drug emissions, and check the whole cell protein in the drug development process is also very important. In the agriculture aspect, ELISA technique is widely used in testing soil, water, seed, compost, biosolid, and plants tissue samples. It also can be used to ensure the safety of the sample against hazardous contaminants such as pathogens, toxins, pesticides, and heavy metals. It is used to evaluate the quality, compositions, and suitability from the fuel output. In the animals and plants disease diagnostic, ELISA is used to detect the animal or plant disease and study the biotic pathogens and host pathogen research. In the food safety field, ELISA is used to check the food labeling, hygiene, additive, and the pesticide residues. Next, I would like to talk about the key parameters or optimization tips for ELISA kit development. These are the key parameters which include sensitivity, accuracy, precision, stability, selectivity, and others parameters such as drug resistance or dilutions, linearizations, and so on. Assay sensitivity refers to the ability of methods or instruments to detect and analyze at a specific concentration and is often defined by a detection limit. The lowest limit of quantifications is the lowest standard curve point that can still be used for quantifications. They are the values above these quantitative result may be obtained with a specific degrees of confidence or the lowest concentrations of an analyte that can be accurately measured. The accuracy of the kit development is also important, and coefficient variation should be plus minus 10% of the theoretically result. For the kit precisions within run and between runs, the CV values need to be within plus minus 20%, and we normally will control it to be plus minus 10% within run and plus minus 15% between runs. In the stability test, reagents used in the assay are thermostable. It is important to validate how long this developed ELISA kit can last and effective. Normally, it can last about 1 year for 80% of the ELISA kits. Selectivity is crucial for ELISA kits development through adding drug sample in the 10 individual source blind control serum to verify the substance to minimize or avoid the interference. If the interference CV values is plus minus 20% for at least 80% of the sample control, then it is considered good. Beyond choosing the appropriate format, each step in the ELISA development is an optimized opportunity. Empirical testing of some of or all of the following are required to get maximum signal to noise ratio. First, variations in the plate chemical may result in the different antigen absorption, efficiency, and non-specificity binding, affecting the signal to noise ratio. ELISA plate geometry can also affect the plating efficacy and the ultimately final signal strength. Side-by-side comparisons of the alternative plate chemistries and the geometry should be performed to determine best practice for your ELISA. Testing a capture ELISA formats is useful to remove the potential epitopes, masking, or conformational change issues. If possible, maintain an optimal temperature, pH, and salt concentration are crucial for optimal performance. These also include binding buffer, coating, and wash buffer as well. When it is in the high temperature condition, then the incubation time will be shortened, else it will increase the non-specific binding. For this reason, room temperature overnight plating and 37-degree blocking, and then after that, room temperature for binding buffer incubation is the most appropriate condition to work with. Again, optimizing the various conditions like temperature, pH, salt, and irrelevant blocking protein concentration will benefit the ELISA kit development. Other than that, ELISA performance is highly dependent on the antibody affinity. Different clones will perform differently. Titrating antibody concentrations and need to be optimized to accommodate for even low affinity antibodies. Negative control is crucial for the kit developments, as increasing the antibody concentration will impose the higher risk of non-specific binding. Selections of what kind of antibodies as capture or detection will also affect the sensitivity of the assay. Monoclonal antibodies offer a specificity. It is typically have a lower signal due to the limited epitope recognition. Polyclonal antibodies may recognize more epitopes of antigens, but with higher unwanted cross-reactivity potential. In sandwich ELISA, monoclonal capture antibody will recover only specific targets from a sample. A polyclonal capture antibody will recover the maximal amount of the target, but with some non-specific binding. Sample quality also play a crucial role in ELISA performance. Complex samples would benefit from the specificity of the monoclonal antibody capture, whereas sample with the simple composition but with low target concentration would be benefit from polyclonal capture. Lastly, the enzymatic signal developments provide robust detection. Time, temperature, and volume need to be optimized prior to the quenching reaction. Short of time with low temperatures may reduce the signal generations. However, long time incubations with high temperature will increase the noise. Volume is limited by the well size, and it must be consistent and optimized throughout the study. While running the ELISA, there are several steps where potential problems can occur and affect the outcome. The most common seen problems are listed here with possible solution. When the low OD or false negative result detected in ELISA test, there are some of the possible reasons may lead to this. Possible is the pipette is not function well and inaccurate. Possible of the incomplete reagent mixing, insufficient incubation time, therefore low or no signal detected. Temperature is too low, inaccurate plate reader filter selections, adding wrong buffer in the process. Poor standard curve is observed due to the improper washing and aspirate step with sometimes cause bubble in the well too. Incomplete reagent mixing and sometimes insoluble particles were found in the reagents control. Inconsistent reagent preparation will also affect the standard curve. Is some of the wells scratched by the pipette tip will also affect the difference of the standard curve. High variations of the CV values in the duplicates occur due to the inconsistent protocol, poor washing step between the duplicates. Human technical error can also play a role, such as inaccurate pipetting technique, splashing the reagents between wells, drying out the wells, and inconsistent sample handling, for example, variations of due to the freeze and thaw cycle among sample and use of different filter setting to start. Plate sample and reagent contamination will also cause the variations. No color developments is caused by sometimes the capture antibodies or antigens does not bind to the plate, therefore nothing to be detected. Incubation time and temperature is not right, inaccurate, or insufficient antibody or substrate added. Incorrect sequence of the reagent added or plate reader filter selection is not correct. Excessive washing will also remove the binding interactions as well. Next, I would like to share with you all about our GenScript capability in helping our clients to customize and develop their ELISA kit. At GenScript, our custom ELISA kit generation service specifically develop kits that enable the reliable detections and quantitative of your unique antigens. We have more than 10 years of kits development experience. Each kit undergoes extensive assay developments and validation to ensure its sensitivity against your desired target. With our extensive optimization process, we ensure that our high-quality custom ELISA kit will perform in your assay. We provide one-stop solution from R&D kit developments, productions, and registrations. The whole process divides into two main parts, which are the antibody discovery part and the kit development part. In the antibody discovery part, we start from the immunogen preparations and animal immunization. After that, perform the antibody generation in different platforms like hybridoma, B-cell cloning, or our advanced beacon platform. When the antibodies are generated, we will perform an affinity ranking, epitope binning, and pairings to characterize the antibody leads that ultimately we will start the kit development. In the kit development part, we are able to start from the R&D kit development stage, further down to the clinical trial sample tests on the ELISA kits of what we develop. And when the clinical trial data is certified, then we will transfer the kit development technique from our R&D site to production site. After that, we can, based on the kit registration and requirement for the FDA standards or based on your government registration standard, to file the kit registration document. When the registration approved, then we will start mass productions of kit manufacturers in GMP level. In GenScript, we offer the service that encompass all the different ELISA formats. Besides then the standard ELISA like a sandwich ELISA, direct or indirect ELISA, or competitive ELISA, we also offer a specialized sandwich ELISA kit development service such as pharmacokinetic ELISA kits, anti-drug antibody ELISA, and whole cells protein ELISA. These kits are tailored more specifically to drug developments and biologics manufacturing. We have developed our in-house antibody discovery technique to improve the antibody generation process, which include we have designed the tools using an advanced algorithm to generate superior peptides antigen sequence. We also invented a proprietary method, ImmunoPlus, to break the immune tolerance of high homologous antigens. We also have a germinal central nuclear protein transgenic spike to enhance the somatic hypermutation and antibody specificity. We also perform the epitope binning to validate the antibody whether they are competing with same antigens epitopes. Lastly, we also have a proprietary adjuvants which will elicit immune response faster to generate large titer of higher specificity antibodies. As I mentioned just now, our GenScript provides us one-stop solutions from the antibody generation to kit development. Start from the initial phase, we will start with the immunogen preparations and animal immunizations, and based on the requirements of the customer and kit's needs, we will perform either monoclonal or polyclonal antibody generation. In monoclonal generation, we have the hybridoma antibody or B-cell cloning methods. In hybridoma platform, we have our proprietary methods, ImmunoPlus and the monoboost technique to break the immune tolerance of highly homologous antigens and to generate a surrogate antibodies. We also utilize the cutting edge of electrode fusions and plasma B-cell enrichment technique to fuse the mouse B-cell with the cancerous myeloma cell to form the hybridoma clones after the perform high-throughput antibody screening. In the B-cell cloning and single B-cell platform, Beacon platform, performed in the rabbits, rabbit generate monoclonal antibody have a superior specificity, affinity, and most diversified antibody to capture all opportunity with high success rate. In this Beacon's plasma B-cell antibody discovery process, which shortened the timeline of typical hybridoma workflow from culture, screening, clonings, and assay detection from originally 12 weeks to 1 day by Beacon machine. We deliver more than 200 rabbit monoclonal antibody projects, and we have 100% immunogen positive delivery rate. We perform antibody sequencing, and we have a professional bioinformatics team to assist you in antibody engineering. In the polyclonal platform, we can provide high-end customized project in different species like rat, mouse, rabbits, or goat. We utilize our proprietary techniques to fasten the polyclonal antibody production in 45 days. We deliver high-quality products in cost-effective values. After the antibody generation, we will go through the screening and characterization step to identify the binding, affinity, specificity, pairings, antibodies clones by using the following assay to check the relative affinity ranking through the biocore or ELISA. Ligands blocking and activity through the IC50, target specificity and species cross-reactivity, epitope binings, and cell-based functional assay affinity measurements and antibody sequencing. And our GenScript have a wide range of characterization screening lists available to satisfy our customer needs. let me share one type of antibody we have done in GenScript. Anti-idiotypic antibodies are the antibodies that bind to the antigen binding site of another antibody or biologics. Anti-idiotypic antibody are important for important downstream reagent for specific drug development assay, which include PK and ADA and others applications such as diagnostic assay and biomarker discovery and evaluation. There are different types of anti-idiotypic antibody binding way. The key success factors in the development is to understand the intended applications and design of proper screening cascade for the selections of ideal anti-idiotypic antibody. There are three types of anti-idiotypic antibodies. Type 1 is the antigen blocking antibody, which bind directly to the antigen binding site of an antibody drug directly compete with the target antigens. Second is the non-blocking anti-idiotypic antibody, which bind to and does not overlap to at all with the paratopes of the antibody drug, allowing the antibody drug to bind to its target antigens and anti-idiotypic antibodies simultaneously. In type 3 is the complex specific anti-idiotypic antibody, which only bind to the idiotypic of an antibody drug when the drug is already bound to its appropriate antigens. Since most anti-idiotypic antibodies are generated against specific antibody drug, they're commonly used in the preclinical practice for pharmacokinetic analysis. Pharmacokinetic is the study of drug metabolism throughout the bodies, and specifically clinicians will determine the rate of the drug absorption, distribution, bioavailability, and the excretion in various cohorts of the patients. In order to accomplish this, researchers need to be able to track the antibody drug which are bound or unbound to their designated target at various time points after post-delivery. Through the use of anti-IDs, primary monoclonal antibodies, various forms of the antibodies therapeutics can be easily tracked and quantified in patient serum, blood, urines, or others body fluid. Other than that, it works as a neutralizing monoclonal antibody was used as a positive control to measure the level of the neutralizing antibodies. Polyclonal anti-idiotypic antibodies are commonly used for immunogenicity assay as reference control. Immunogenicity is the ability of a therapeutic such as antibody drug to induce a humoral or cell-mediated immune response, which lead to development of an anti-drug antibody. Therefore, it is extremely important for researchers to analyze the immunogenicity of a new antibody during the preclinical assays. An anti-drug antibody is very similar to an anti-idiotypic antibody since both bind to the same antibody drug. Therefore, a pool of the polyclonal anti-idiotypic antibody can be used as a positive control when analyzing for the presence of anti-drug antibody within the patient samples. This is anti-idiotypic blocking antibody generation cases from our GenScript. And the drug FA2 immunogens were injected into the rabbits. From one round of the B cell cloning, we are able to obtain a 154 antigens positive clone, and human IgG1 was used as a negative control. 81 clones are further studies, and then the 81 clones are very sensitive to the FAB2 and the full-length drug antibodies. We can see that the 81 clones have OD level bigger than 1 for the FAB2 and intact full-length drug antibody, and the OD level is less than 0.2 for the total human IgG negative control. Positive control and negative control are included, and the OD is larger than 1 and lesser than 2 respectively. Next, we carry out the blocking antibody screening. The blocking antibody screening methods of the anti-idiotic antibody shown here. In the blank sample, the antibody drug is coded, and then the target is labeled to detect the signal. At this time, the target can be bind to the antibody drug, and the signal is also very high. When it is a blocking sample, after the blocking anti-idiotic antibody is added, the target will not be able to bind to the coded antibody drug, so it will be washed away during the washing step, and the part or all the signal cannot be detected. In addition, the screening of the blocking anti-idiotic antibody can also be achieved by coding the target and label the antibody drug for detections. After the blocking screening was done, then 10 clones with the blocking rate bigger than 80% were obtained. So after the discussion of how our GenScript to perform the antibody drug discovery part, now I shall focus on the ELISA kit development. This is the overall workflow of the ELISA kit development. First, we will proof of the concept in 1 to 2 weeks times. In this step, we start with the identifying the best pair of antibodies and evaluate the performance in sensitivity and specificity checking. We perform the optimize the dilution, concentration, and serum validation based on the customer needs to establish the standard curve and evaluate the consistent data from the customer organizations. Second step is to perform the assay development. Based on these pair antibodies, we have to optimize the parameters and condition, for example, coating concentration, detections, reagent concentration, dilutions, and blocking buffer optimization as well. We will evaluate the specificity of the antibodies by testing different concentrations of similar target to understand the interference levels. In the third stage, we will perform an assay validation, which requires 3 to 4 weeks time. We will manufacture 5 kits for assay validation to check the stability, accuracy, and the variations of the assay kits. For the criterion of the assay kits, we will make sure the intra-assay precision is less than 10% and the inter-assay precision is less than 15%. And the recovery range is plus minus 15% according to the FDA requirements. After all these pass, then we will perform the kit manufacturing within which require 2 to 3 weeks. Customer can order start from 5 kits. In normal drug discovery development process, PK, ADA, HCP, ELISA kits are commonly used. In ADA ELISA kit development, we normally require 13 to 19 weeks. In HCP ELISA kit development, we require 10 to 15 weeks. For PK ELISA kit development, we require 9 to 13 weeks. Now let's see how does PK kit develop. ELISA methods and magnetic particles-based chemiluminescent immunoassay are both suitable for PK kit development. And there are 2 main types of PK kits. One is sandwich and another one is competitive. Sandwich ELISA kits and the magnetic particles-based chemiluminescent assay immunoassay are commonly used in the macromolecules drug PK testing, for example, antibody drug. This is because there are multiple surface epitopes for different binding targets. Capture antibody can bind to the epitope 1, and the detecting antibody can bind to the epitope, another epitope, and then this will with a good pairings. If we conjugate it with the capture antibody on the magnetic bits, and then detecting the antibodies was read by the chemiluminescent, then this will be the magnetic particles-based chemiluminescent sandwich immunoassay. However, due to the small surface areas and the limited epitopes of the target antigens for the small molecules drug, for example, kinase inhibitor, it is hard to find a good pairings antibodies for this kind of assay. Therefore, competitive PK assay is suitable for this kind of studies. When we coated the small molecules drugs on the plate and put in the enzyme conjugated antibodies or non-conjugated antibody to compete, then we will see the color changes. This is similar to the magnetic particle base as well. This is one of the PK sandwich ELISA developed by our GenScript. This kit we have developed met and exceed the FDA requirement. The precision of the intra and inter-assay variations are both less than 5%. For FDA requirements are less than 20%. We are actually able to meet and the accuracy and recovery as what FDA require. ADA drug validation is important in the drug discovery and development state. When drug is taken and going into human body, sometimes body will create the antibody to neutralize this drug antibody, then which will inhibit the drug activity and reduce the efficacy. Other than that, sometimes it will cause anti-drug antibodies immune response, which will pose a danger to the lives. According to the FDA guideline, which recommend that drug immunogenicity should be monitored and tested in preclinical, clinical phase 1, 2, 3, and post-marketing to ensure the drug safety. In the ADA validations assay development, we perform direct or indirect ELISA. If an antibody drug induce the developments of anti-drug antibody, they can be detected through direct binding ELISA. In this assay, an antibody drug is coated on the plate and then is coated that on the plate is washed with the serum, and then the plate is then washed with label IgG, which only bind to the constant regions of the anti-drug antibody. Therefore, the ELISA will only show fluorescent if there are anti-drug antibodies present in the sample serum. In the second type, which is the bridging ELISA sample, an antibody drug coated on the plate is exposed to the serum, allowing the anti-drug antibody capture. The same antibody drug but label is used for detections of the anti-drug antibody through the bindings to their SCFV regions. Both of these assays need to be analyzed via a positive control, such as anti-idiotic antibody. Whole cell proteins are those produced or encoded by the organism and unrelated to the intended recombinant productions. HCP will induce anti-HCP antibody that can induce a clinical effect in patients. In addition, HCP may possibly act as adjuvants which can induce the anti-drug antibodies that can affect the safety and efficacy of the drug. Therefore, researchers have to detect HCP during the preclinical lots using the toxicology assessment, or lots during the clinical developments, and the process validation sample from the final manufacture process. This is the workflow for HCP kit development. First, we will extract the empty whole cell as an antigen and prepare the high molecular weight and low molecular weight whole cell protein as an immunogen. Before immunization, we will screen the serum of the animal model to check and confirm the animals are not contaminated. After that, we will immunize 8 rounds for the high molecular weight proteins and 10 rounds for the low molecular weight proteins separately, as low molecular weight proteins is less sensitive to be detected. Therefore, this will increase the immune response. Every 3 rounds of the immunization, we will perform an indirect ELISA to check the immune response. After that, we will collect the blood to perform a 1D Western blot to test with the immunogen. After that, we will run a 2D SDS page to confirm and find the optimal mixing ratio according to the FDA guideline and ultimately for the kit development. This is how HCP ELISA kit development process. Before HCP kit develop, we need to prepare the HCP for antibody generation. HCP are extracted and separated into high molecular weight and low molecular weight for immunization. After the antibody were generated and purified, and we have to make sure the purity is more than 90% and the ELISA titer is more than 512K, and the coverage of the SDS page and 2D Western blot is more than 70%. We mix the high molecular weight and low molecular weight protein generated antibody in different ratio to identify the best combination ratio for total HCP detection. This is the example of 2D Western blotting of the HCP with the different ratio of the high molecular weight and low molecular weight antibody. We can observe that 1:1 ratio of high molecular weight and low molecular weight antibody have a better coverage. This is one of the example of our in-house generated HCP ELISA kit case. In this ELISA application, we would like to detect the CHO protein, as we know that CHO cells are widely used in the therapeutic drug development. That this is to test whether there are any residues remain in the drug. If the poly antibody in the sandwich ELISA set out, the linear range is 1 to 100 nanogram per ml, and the sensitivity is 0.608 nanogram per ml. The intra-assay variation is less than 5%, and the inter-assay variation is less than 10%. Recovery is around 85 to 115%. Based on our developed HCP ELISA kits, we are able to hit within the range of the FDA requirements. This is another type of HCP sandwich ELISA case developed in our GenScript for E. coli whole cell proteins detections. The linear range of this is around 0.781 to 15 nanogram per ml. Sensitivity is 0.807 nanogram per ml. And the intra-assay variations and the inter-assay variation are both greater than the FDA requirement standard, which are less than 8.44 and 10 respectively. The recovery range is within the FDA requirement from 80 to 120%. Besides the PK and ADA and HCP ELISA kit developments, we also provide customizations for others ELISA kits to detect biomarkers. In the case study 4 of the Bama miniature pig serum detections and the case study 5, which is the rat serum, we are able to meet and exceed the FDA requirements, and normally the intra and the inter-assay variation of our generated ELISA kit are less than 10%, which is less than the FDA requirement, less than 20%. This shows that our customized ELISA kit is produced in very high quality and high technique. In summary, we are expert in customized antibodies and ELISA kit service. We deliver customized ELISA kit with high quality. We are the award-winning customized antibody suppliers. We have followed the stringent clinical FDA grade development standard. Our kits release above FDA standard. For example, the kit recover is between 80 to 120% and the precision CV is less than 10%. We have more than 70 years antibody discovery experience and deliver 70 kits projects with more than 150,000 kits. We provide one-stop solutions from research grade to GMP level to match different application requirements. That's all for my today presentation. If you have any inquiry, please feel free to contact me through this email. Thank you for your attention.